Good evening, everyone. At present world, we are facing a global health crisis. But the solution isn't in a lab, it's hidden in nature. Today we are exploring our presentation on metagenomics in drug discovery, unlocking the nature's hidden antibiotics. Here I am Farhatul Hasan Shatu from the Department of Pharmacy at Jashore University of Science and Technology, and I will be guiding you through this session. Before we start, let's look at the core challenge. Did you know that over 90% of the microbes in the world can't be grown in a petri dish? Yet these uncultured microbes hold the genetic key to the next generation of antibiotics. They are nature's hidden treasure chest, and traditional science has largely been unable to open it. This failure of traditional science is happening while the threat of antibiotic resistance continues to grow. The stakes are incredibly high. The antibiotic resistance crisis is projected to cause 10 million deaths annually by 2050. Traditional methods are simply recycling old compounds, and we are running out of options. We desperately need novel antibiotics, and this is exactly where metagenomics steps in. So how exactly does this metagenomics solve the problem of unculturable microbes and leads us to new drug? Metagenomics is the study of genetic material taken directly from the environmental sample like soil or water without needing to culture the microbes first. And here we bypass the whole 90% problem. It's basically used to main a process. First one is sequence based, where we look for antibiotic producing gene, and the second one is function based, where we screen the actual DNA fragments for the desired bacterial activity. This is the modern way to search for life-saving drug. Let's look closer at this process. How do we go from a bucket of dirt to a potential new drug? The process is systematic. We start with sample collection, perhaps a handful of dirt from a remote location. We then extract the total DNA from everything in that sample. This DNA is then broken down and sequenced. And finally, powerful computational tools piece together the data to predict and analyze potential new functions, ultimately leading us to a novel antibiotic. Here we are just not talking about theory. The field has already produced revolutionary discoveries. The first breakthrough was teixobactin, discovered from uncultured soil bacteria using the innovative ICIP technology. It was effective against highly resistant pathogens. Another example is carbamazepine, the first antibiotics discovered directly from a metagenomic library. This success highlights the advantage of metagenomics, but like any cutting-edge field, it comes with its own set of challenges and clear directions for the future. In summary, the key advantage is the access to the 99% of uncultured microbes, dramatically expanding our discovery pool. However, there are challenges mainly around gene expression. But the future directions are exciting. We are moving toward metagenomic guided synthetic biology and exploring extreme environments for unique compounds. At last, I will leave you with this final thought that the next miracle antibiotics isn't in a lab, it's hidden in nature's genome. Metagenomics is simply giving us the lens to see it. Thank you, everyone.